Hello everyone, welcome back. Today we're going to be moving on and getting into something that's a little more complex. We're going to take the next step into genetics, moving past Mendelian genetics, looking at non-Mendelian genetics, which is really just more of our modern understanding. It's everything we've learned since Mendel, last 200 years since Mendel, with increasing technology. It does get a little more complex, right? But that's just the reality of genetics and inheritance. But before we get into this today, we are assuming, with some of our key vocabulary here, that you have already watched and gone through our, classical traditional Mendelian genetics content so far. We're going to be assuming you know some vocabulary like zygosity, like if you're heterozygous or homozygous and what exactly that means. We're also going to be assuming you have at least a basic understanding of the patterns of dominance that happens between the interactions of alleles, like complete dominance, codominance, and incomplete dominance. That'll be coming up today as well. So if you haven't done that, definitely go back and watch that first. I'll be meeting you back here. So as soon as we cross this threshold into our more modern understanding of genetics, which we're just going to refer to as non-Mendelian genetics, we're very quickly going to arrive at the idea, the concept of multiple alleles. Now, multiple alleles is kind of exactly what it sounds like, right? In our traditional Mendelian traits, we have one gene, one trait, and that gene is composed of two alleles. One we inherited from our biological mother, and one we inherited from our biological father, right? Mom and dad. We get one from mom, one from dad. So there's like copies of all of our genes, right? We call those copies or versions of genes alleles. But now we're going to start looking at genes that have multiple alleles that are possible to inherit, right? So we're really working with about 3 or more alleles, right? It can get really complex. But today, for the example we'll be working with today, we're really just looking at 3. So it's multiple alleles, at least 3, all working within a gene for one trait. So that's what we're working with there. The next thing we see is, within these multiple alleles, is that each of those alleles can have a different relationship with each other, meaning we can see the different patterns of dominance emerging between the different alleles. So for example, allele 1 could be completely dominant to allele 2, whereas allele 1 is maybe co-dominant with allele 3, right? Like 2 different relationships between the alleles. And maybe allele 2 is incompletely dominant to allele 3. Who knows, right? We could see any possible combination of those different patterns of inheritance and different patterns of dominance working on these multiple alleles. So let's get to our example, right? Let's actually make something concrete here to help clarify and maybe visualize this a little better. We got a pretty big one. It has a lot of implications. Today we're going to look at one of the genes involved in human blood type. It's called the isoagglutinin gene. It's just a protein in your blood, right? It's used in determining your blood type. Blood type is actually polygenic. There's actually a polygenism there. That's for next time. That's the next step into non-Mendelian genetics. But for today, we're going to look at the one gene, and there are multiple alleles in this gene for blood type. You've probably heard of them before. They're commonly referred to as allele A, allele B, and allele O. Right? Think of the blood types, right? A, B, and O, and AB. We'll get into that. So we have these 3 alleles here working in blood type. Big example, has a lot of implications. This comes up in the medical field, right? Like doing blood transfusions. Huge deal to know somebody's blood type. It also comes up in forensic science, or just name a couple examples. In forensic science, right, if you're finding blood on a crime scene, you're going to want to type it, see if you get a match to any of your suspects, right? So it's pretty good to have a basic understanding of blood type and how it works, where it comes from. So we have 3 alleles here, A, B, and O. Let's get into their relationships, right? How do they interact? How do the different patterns of dominance work between the 3 alleles? So first, A and B. A and B are always completely dominant over O. Right? O here, the O allele, is always recessive. It's like, okay, that's pretty Mendelian, right? That's pretty traditional. We've seen complete dominance a lot in that case. But the interesting thing here, the second relationship we're going to look at that stands out and makes human blood type unique, is that A and B, when they're together, they are co-dominant to each other. Right? And that's where the blood type AB comes from, right? Blood type AB, both alleles being expressed together at the same time. We're going to see them both. So those are the rules that this gene and these multiple alleles are going to follow. Let's just move on here a bit. Look at this breakdown. So following these rules, we can start to get into, okay, what's our genotype combination that's going to give us the phenotype of the blood type itself, right? There's a couple possibilities based on these different patterns of dominance that are interacting between the different multiple alleles. So as you see here in this chart, we got genotype going on the left-hand side, the two genes we're going to inherit, right? One allele from mom, one allele from dad. Those are two alleles. And the phenotype, the way that these alleles that are inherited actually get expressed, going through protein synthesis and gene expression. So let's start with blood type A. Two possibilities here based on the patterns of dominance, right? We could be, number one, we could be AO, right? A is always completely dominant to O, so if you have at least one A, it'll mask the O, you'll see the A there. Or you could be homozygous dominant, right? You have two copies of A, right? We have so a heterozygous option and a homozygous dominant option. Moving down to blood type B, it's going to operate very similarly, right? We could get B, and we could get an O. We could have BO, right? Could get BO in there. Again, that's heterozygous. B is completely dominant to O. We could also be homozygous dominant, right? Two copies of that B allele. Definitely going to be blood type B there. Blood type AB, the third one here, only one way for that one to play out, right? So when A and B are co-dominant to each other. So the only genotype option there is AB. And O, the last one, always recessive, right? So the only way to have the phenotype of blood type O is to be homozygous recessive, two copies of the O allele. So that's where this is coming from. That's how this breaks down, right? Your genotypes, your phenotypes, and how we see the different patterns of dominance interacting between the multiple alleles. So there's that chart. Quick erase here. I'm going to move on. So we can also predict here, right? We can actually go back to our other old friend, the Punnett square, and we can use the Punnett squares to predict offspring based on known phenotypes and genotypes for blood type. So coming up to our worked example, we know, and again, this is just an example I'm making up, right? Just an example we'll work with here today. Let's say that I know I am heterozygous for blood type A. Heterozygous for blood type A. And let's say I am going to reproduce with a partner that I know is heterozygous for blood type B. What are our children going to be, right? What are these offspring going to look like in terms of their blood type? So we can figure this out just based on the information we have here, right? So then you scroll down a little bit, bring this Punnett square up. So let's start setting up our Punnett square. Let's figure out this cross. Start with me. I know my blood type is heterozygous A. Hetero means I need two different alleles. Only way to have two different would be blood type A. That means I have to be AO. And my reproductive partner in this case, also heterozygous blood type B, they have BO. But I don't mind, right? We're reproducing here, so it's all good. So we got our Punnett square, right? Basic Punnett square set up. I'm just going to take my genotype and split that up across the top. So there's my A allele, there's my O allele. I'm going to take my reproductive partner's type, the BO, split them up down the side. Setting up the Punnett square. Again, kind of assuming you've already gone through this before, but here it is again. Applying it with multiple alleles. All three alleles in play here. So we fill this in, we do the cross. We're going to get AB there, AO here, BO over there, and OO down there. Right, just kind of filling out the Punnett square here, right? Making sure all of our alleles are lining up. And right off the bat, notice that's a lot of genetic diversity, right? There's actually a 25% chance to get each of the different genotypes and each of the different phenotypes, right? We have 25% of our offspring are going to be blood type AB. Another 25% are going to be blood type B heterozygous. Another 25% going to be blood type A heterozygous. And finally, we got the homozygous recessive blood type O. So we actually see every blood type emerging in that F1 generation, right? That first generation of offspring. We see every possible blood type there coming from me as one reproductive partner and this other heterozygous B reproductive partner. So that's what you can predict, right? Seeing where these multiple alleles are going to play out here. So another quick erase. And then we're coming up to the practice problem we have for the send-off today. So just like, so here's a little bit of the answer explained, right? I put it into type here. Essentially, it's a 25, 25, 25, 25% breakdown, right? It's a 1 to 1 to 1 to 1 ratio in that Punnett square. And we got some more practice, right? We can really start answering some very almost like fertility clinic kind of like questions, right? And another application of understanding multiple alleles in human blood type. So here's a question, and definitely flush this out and support this and back this up with Punnett square. Definitely show your work and support it here. But the question is, a man with type AB blood, right? Blood type AB. Is it possible for him to be the father of a daughter who is blood type O? So you might think you can just figure that out just based on your definitions, your key terms, your vocab, your understanding of the concept here of multiple alleles, but also take it a step further, support the work with Punnett square. See you next time for the solution. Good luck.